Gene therapy is an innovative approach to treat inherited diseases by targeting the root cause at the genetic level. In this video, we'll explain how gene therapy works and highlight its potential in treating conditions such as cystic fibrosis and muscular dystrophy. Gene therapy works by repairing or replacing faulty genes in the cells of individuals with inherited disorders. Imagine it as fixing a malfunctioning part in a machine. By inserting the correct gene, the body begins to function properly again. Gene therapy shows great promise for treating cystic fibrosis, a condition where thick mucus builds up in the lungs due to a faulty CFTR, or cystic fibrosis transmembrane conductance regulator gene. By delivering a healthy copy of this gene, patients experience improved lung function. Muscular dystrophy is a progressive genetic disorder that primarily affects the muscles, causing them to weaken over time. The condition is caused by mutations in the dystrophin gene, a gene responsible for maintaining the structure and function of muscle fibers. Without a properly functioning dystrophin gene, the muscles are unable to repair themselves, leading to a gradual loss of strength and mobility. However, gene therapy offers new hope. By introducing a healthy version of the dystrophin gene into muscle cells, we can potentially restore their ability to function properly. This means that with successful treatment, the muscles may regain strength, allowing individuals with muscular dystrophy to maintain better mobility and a higher quality of life. There are two primary methods for administering gene therapy: in vivo and ex vivo. In vivo involves delivering the healthy gene directly into the body, while ex vivo entails removing cells, correcting them in a lab, and reintroducing them into the patient. While gene therapy holds immense potential for treating a wide range of inherited and acquired diseases, it still faces several significant hurdles that must be addressed to maximize its effectiveness and accessibility. One of the most pressing challenges is the body's immune system. Gene therapy often involves using viral vectors, modified viruses designed to carry healthy genes into specific cells. Although these vectors have been engineered to be safe, the body's immune system may still recognize them as foreign invaders and initiate a defensive attack. Finding the right balance between ensuring safety and enhancing the efficiency of gene delivery is an ongoing area of investigation. Ensuring that gene therapy reaches all those in need, regardless of socioeconomic background or geographical location, is critical for the future of this promising technology. Gene therapy has made remarkable progress with the help of new genetic editing tools like CRISPR, clustered regularly interspaced, short palindromic repeats. CRISPR is a revolutionary technology that allows scientists to edit genes with unprecedented precision. Unlike traditional gene therapy, which involves adding healthy genes to replace faulty ones, CRISPR goes a step further by directly targeting and repairing the defective part of the gene responsible for causing a disease. This level of precision means that we can correct the exact mutation at the DNA level, preventing it from producing faulty proteins and restoring the body's normal functioning. With CRISPR, the potential for treating genetic diseases is vast. Inherited conditions like cystic fibrosis, muscular dystrophy, and sickle cell anemia, which are caused by specific genetic mutations, could be addressed by precisely correcting these errors. Though still in its early stages in clinical applications, CRISPR's ability to fine-tune genetic corrections means that treatments are becoming not only more efficient, but also safer and more tailored to the patient's unique genetic makeup. As a result, the future of gene therapy is growing increasingly hopeful, with CRISPR at the forefront of these exciting developments. There are several ethical concerns that arise when we discuss gene therapy, particularly when we consider its long-term implications. One of the most debated aspects is the idea of altering germline cells, the genes that can be passed down to future generations. While such advancements hold the potential to eliminate hereditary diseases entirely, they also raise questions about the unintended consequences these changes could have on future individuals and society as a whole. Should we be allowed to make changes to the human gene pool that could affect not just the current generation, but many generations to come? Beyond the ethics of genetic alteration, there's also the critical issue of accessibility. While gene therapy holds incredible promise, it's important to ensure that these treatments don't become exclusive to those who can afford them. Making sure that gene therapy is accessible to all, regardless of socioeconomic status or geographical location, is crucial if we are to truly harness its potential for global health. After all, every individual, regardless of background, deserves access to life-changing medical advancements that can vastly improve their quality of life. Gene therapy is revolutionizing the medical field, providing a beacon of hope for millions suffering from inherited diseases. As this technology progresses, we are witnessing significant advancements not only in the effectiveness of treatments, but also in the potential to make these life-changing therapies accessible to all. Over time, we can anticipate even greater breakthroughs, bringing us closer to eradicating the root causes of genetic disorders. As we move forward, it's essential to continue investing in research and ensuring that these treatments reach every corner of the globe. The future of medicine lies in this delicate balance between innovation and inclusivity, offering the promise of healthier and brighter lives for future generations. Gene therapy, transforming lives.